Hi, I'm Melba Priyadharshini, Assistant Professor, Department of Biotechnology. Today we are going to discuss about plant tissue culture. Definition of plant tissue culture. Plant tissue culture is a collection of techniques used to grow plant cells, tissues and organs under the sterile condition on the nutrient culture medium. And it is a vital tool in plant biotechnology, horticulture and conservation. History of plant tissue culture. Although tissue culture has been around since the beginning of 18th century, plant tissue culture only began in the year 1898. And Gottlieb Haberland, he is a German botanist, made the first attempt to use the in vitro method when growing plant tissue culture. The cells he used were varied palisade tissues coming from the leaves, pits, epidermis and epidermal hairs. And applications of plant tissue culture. It is used in the development of disease-resistant crops by using their meristem, that is apical part of the plant, that is named as an explants. Tissue culture is also used to develop thousands of genetically identical plants from one single parent plant and rapid propagation of economically important plants. It has been used extensively for the production of commercially important plants including food plants like tomato, banana, apple, etc. And conservation of endangered plant species can be done through tissue culture techniques. And the laboratory system, that is set up and aseptic techniques. Okay. So what are the essential equipments needed in the laboratory? One is laminar airflow chamber, autoclave, pH meter, incubator and some of the culture vessels are required. Sterilization method. Autoclaving to 121 degrees centigrade for 15 to 20 minutes and surface sterilization using chemicals. This surface sterilization is done for the explants using the ethanol, sodium hypochlorite, mercury chloride, etc. And UV sterilization process is followed in the laminar chamber. And media preparation and dissolving the desired that is nutrients media is dissolved in the distilled water and it is adjusted to the pH of 5.7 to 5.8 and it has been autoclaved. Medium. What is medium? A plant tissue culture media is a combination of nutrients and regulators that help plant cells, tissues and organs to grow and multiply. Composition of the nutrients. Okay. Composition of the media, they are macronutrients, micronutrients, vitamins, carbon sources, solidifying agents. Okay. What is macronutrients? Macronutrients are maximum amount of nutrients present in the media. The macronutrients provide the 6 major elements, that is nitrogen, phosphorus, potassium, calcium, magnesium and sulfur required for the plant cells or tissue growth. The optimum concentration of each nutrient for achieving maximum growth rate varies considerably among the species. And micronutrients are minimum amount of nutrients required. Micronutrients are essential elements that plants need in small amounts for growth, development and reproduction. Iron, zinc, copper, boron, etc. And vitamins like thiamine and pyridoxine and carbon sources such as sucrose is required and solidifying agent agar. And there are different types of medium and this media is based according to the species of the plant. And the types of media are MS medium, which is called as Murashige and Skoog medium, medium Lensmeyer and Skoog, and medium Gamboge, Nist and Nist medium, White's medium. And commonly used media are Murashige and Skoog medium, which is known as MS medium, and Gamboge B5 medium. Plant growth regulators, which we call it as the hormones for the plant. So main growth regulators are auxins, cytokinins, gibberellins, ethylene and ABA, abscisic acid. So what are the auxins? Auxins are helpful to promote the rooting and cell elongation. And example of auxins are IAA, that is indole acetic acid, and NAA, naphthalene acetic acid. And cytokinins are useful to promote, that is stimulate the shoot formation. Example, BAP and kinetin, that is benzyl aminopurine and kinetin. And gibberellins are helpful to promote elongation of internodes. And ethylene and abscisic acid plays a role in stress responses and embryogenesis. Techniques in plant tissue culture. Okay, there are, that is 3 types. Let us discuss some of the types here. Callus culture. Callus and undifferentiated mass of cells with plant regeneration potential and can be grown in labs from various plant tissues. These cultures require subcultures every 28 days and vary in structure and growth based on the plant sources, nutrients and the environment. And organ culture. Organ culture can be defined as the organs or plant parts culturing in an artificial media or culture from isolated medium. Any part of the plant can serve as explants in organ culture like shoot, root and flower. Here you can see the structure, that is callus culture and organ culture. From the plant, a part of the plant is chosen, leaves, and it has given a proper medium. After callus, the callus has been propagated into shoot and root and then taken to the greenhouse. Next is micropropagation. Micropropagation is the method of plant propagation, usually using extremely small pieces of plant tissues taken from a carefully chosen and prepared mother plant. Mother plant is called as an explant, and growing these under laboratory conditions to produce new plants. And there are stages of micropropagation. Stage 0, stage 1, 2, 3 and 4. And the method involves in stage 0 is selection of the explanter. The plant which has to be the plant has to be grown in the laboratory has been chosen. Stage 1 is culture initiation and establishment. Stage 2, shoot multiplication. Stage 3, rooting of the shoots. And stage 4, transfer of the plantlets in the greenhouse environment. Stage 0. This is the initial stage of micropropagation. The stock plants are selected and grown under controlled condition before using them for culture initiation. We have to select a particular species of the plant for micropropagation, and they has to be in controlled condition before the culturing of plants. Stage 1. The explants are established in a suitable culture medium, that is suitable MS or Gambox medium. This stage involves the following steps. That is isolation of the explants. We have to choose either plant parts, that is either it may be a leaves, stems, root or shoot or axillary part. So, and surface sterilization of the plant should be given. As I said before, we have to give surface sterilization for the explants using certain chemicals, and washing the plants with the distilled water. The explant is established on an appropriate culture medium. After the surface sterilization, after washing, the plants has to be given a proper culture medium and it has to be inoculated. And stage 2. This stage involves the rapid multiplication of the shoot or rapid somatic embryo formation in a defined culture medium. According to the medium we have given, the multiplication starts, that is propagation of the plants starts to begin in the stage 2. And stage 3, in this stage, the shoots are transferred to the medium for the development of the roots. Okay, so we have to choose either the cytokinin or auxins. Okay, so the medium has been given the right auxin and cytokinin for the development of the whole plant, or only the shoot the cytokinin has been given if only the root the auxin has been given. So we have to choose either one of the auxin or one of the cytokinin, or only one auxin or only one cytokinin. So the shoots are either transferred directly in the soil for the root development or transferred to the nutrient medium in the laboratory. Either it may be given transferred directly to the soil, or else we can give another medium, that is we can subculture to the another medium of rooting too. And stage 4, in this stage the plantlets are established in the soil. The shoots from the laboratory are transferred to the greenhouse under controlled condition of temperature, humidity and light. Okay, so proper, that is greenhouse, we have to transfer those plants into the greenhouse and then finally to the outer environment. And these are the stages of micropropagation. First we are initiating choosing one X plant and taking one part of the plant and culturing with a suitable medium, and those cells are multiplied, that is micropropagated, and it forms a rooting if we give a proper hormones of rooting hormones, and then shooting with the shooting hormones, and finally a plant. This plant has been taken to the greenhouse. Thank you.